So yeah, the nervous system. First, we're going to talk about its general structure, right? Which means we have two parts, central and peripheral. Okay? So what comprises central nervous system? It's a brain cortex, okay? So brain is large. Most parts of the brain are out of our conscious control. So cortex is the part that is responsible for our conscious ideas. You know, when we consciously make a move, when we consciously appreciate the sensation and the decisions that we make, all the thinking, it's cortex. Cortex is effectively the part of the brain. There are certain parts in the brain that you don't have a control over, but they are indispensable for your conscious activities. For example, I mentioned thalamus. It's the part of the brain that relays the information. So it sends the information, like visual information, to visual cortices, to hypothalamus and other parts. Okay, so it's important. I usually isolate brainstem. This is the typo. So some people consider brainstem a part of the brain, which is not wrong, but I like to specify that brainstem is responsible for all your physiological activities. And your physiological activities, physiological processes are connected to your conscious thinking, right? If, I don't know, like, if you see a dog and you decide to pet a dog, okay, your heart rate will go up because you like this process. Make sense? Or you see something scary and it's scary for you, maybe not for someone else, and again, you develop that sympathetic response. Or you see a picture of pizza and you start salivating in response to that because you like pizza. So those kinds of involuntary activities belong to brainstem. And then we have spinal cord. It's the place for transmitting information, okay, and for a bunch of repetitive movements and reflexes. So far, does that sound good? Now, peripheral nervous system can be divided to sensory and motor. So sensory, obviously responsible for, well, sensing, yeah. So, another term for sensory is afferent, okay? Sensory equals afferent. And motor, another term is efferent. Okay, think about it. I suggest to think about it in the following way. If you are affected by something, it's because you had some sensations about it. Affected. If you want to cause some kind of effect, you need to do things. You need to produce something, to move. Does that make sense to you all? Excellent. Within the motor system, we have two sub-branches, somatic and autonomic. Somatic, motor responses, and we'll get to motor responses in a minute. Somatic motor responses are produced by your skeletal muscles. Therefore, they can be controlled. Autonomic responses are produced by your cardiac muscles, smooth muscles, or different glands, and cannot be controlled. Cannot, no. You will see that we actually have very limited amount of control over our bodies. Okay? I mean, personal control. I'm not talking about government. Autonomic nervous system is divided into sympathetic and parasympathetic branches. Sympathetic division, also known as the producer of fight or flight responses, and parasympathetic is known as the producer of rest and digest. Okay? So now let me sort of step back and illustrate one of the fundamental concepts in the function of a nervous system. If you want. Fundamental concepts in the function of nervous system, specifically how the signal goes through. So here's what I'm going to do. The blue color, the light blue, is going to be sensory stuff. Okay? That makes sense? So everything that's going to be in blue is going to be about sensory. Green stuff will be integration stuff. Like things that happen in your, like, okay, I tell you, write this down. No, it's an example. It's an example. No, it's an example. For example, I tell you, write whatever I'm going to say down. You hear it. That's the sensory input. Then you make a decision. That's the green stuff. Integration. You make a decision. Am I going to write things down? Maybe I'm going to type it down. Okay? And then you produce a response, which is writing, typing, or you say, screw you. Right? I'm not going to do anything. So the response, we're going to put it in red color. So motor stuff. Here's the fundamental thing about nervous system. Let's say you have some kind of a stimulus. I'm trying to find some extra color. Okay. Stimulus is going to be brown. Right? So I have a stimulus. Like I told you something. How do you know that there was a stimulus? You have sensation. Hearing, right? So you have a receptor that receives the signal and transmits the signal through the sensory or afferent fiber. The term fiber, I don't really like it, but the term implies, like, it's kind of nerve. You can use a nerve, you can use it for a neuron, okay? And that sensory signal, remember, blue is sensory, reaches your central nervous system, okay? This is where integration happens. Now listen. Can it be brain? Can it be brain? Is brain a part of CNS? Yeah. So, when I tell you, write something down, is your response voluntary? Yes. That's brain. Now, let's say I tell you, by the way, your next exam is next week. Is your heart rate going to go up? Yes. Is this voluntary? Yes. No, you did not tell your heart to start beating faster. It just, okay, so brainstem can produce a response like that. Now, imagine I'm taking this bottle and throw it at you. You're going to immediately dodge it or something like this, right? You're not going to even think about it. So this reflex, this response is produced by your spinal cord. You can do as well, right? Or let's say you step on the nail. What you going to do? You're going to, well, yeah, but are you going to go on another foot? You're not going to think about, oh, I need to go on another foot because it hurts so much, right? Just going to withdraw your damaged, injured foot. So, spinal cord. My point is this. Your integration centers, whether they are in the brain or they are in the brain stem or in the spinal cord, will produce a response. And no matter what the response is, we consider it motor. Motor does not necessarily imply movement that is visible. So this motor response goes through the motor or efferent fibers until it reaches no, that's not a good idea. The effectors. Do you follow? Effector is something that actually does things. So there are two types of effectors, as we just discussed. Somatic and autonomic. Another term for autonomic is visceral. You know the verb to eviscerate? It literally means to take the organs out. Like, we use it in a different context, but it literally means viscera is everything that's inside of your body. Okay? Like liver and spleen and whatnot. Does that make sense? So the only somatic effectors are skeletal muscles. Clear? Your autonomic receptors can be smooth muscles, cardiac muscles, or glands. Does that make sense? So, you can have let's say you're a normal person and you see some absolutely disgusting Japanese manga and you think, oh God, this is disgusting, like you repulsed, like, yeah. Now imagine you're a pervert and you see like, oh, this is cool, okay? These are your conscious responses in the head, but they will produce involuntary autonomic responses like, I don't know, constriction of the pupils, dilation of the pupils, or maybe your heart rate will go up or down and your palms will start sweating. Sweating is the glandular response, right? So my point is, this, you have to think about the flow of information in the nervous system from that perspective, that it's always an input, integration, and some kind of a response at the end. We good? Okay. By the way, we will figure out why the information flows only one way. So now let's chat about these cells. So these are glial cells, and I genuinely don't know what was in my head when I did not connect individual images to the names. This is what I'm going to do now. So these guys are astrocytes. They are present in the central nervous system. They have two main functions. One of them is to maintain the ion concentrations around neurons. So they pretty much like a stage crew for Metallica. Remember that example from tissues? And they also provide blood-brain barrier, which is super important. You can see how they sort of wrap the blood vessels. So they control very, very tightly what can get from the blood vessel into the brain, because brain is kind of important. Okay? Does that make sense? So it's a very selective barrier, which also is a bit of a pain in the ass for drug companies, because delivering the chemical in the brain is super hard. Okay? Then we have microglial cells. They also present in the CNS only, if it says where. That, by the way, you don't need to know this. I'm not asking any of this. If you're curious, please, but I'm not asking. The diseases, God forbid. So microglial cells are basically macrophages, cells that eat other cells. They provide immune protection, but they kind of suck because they sit in one place. So if something like bypasses them, okay. These are ependymal cells. In observable future, we're not going to talk about them, like we are going to talk about them at some point, but all you need to know is that they produce cerebrospinal fluid and separate blood from this fluid. And you have super important Schwann cells, not Schwann cells, oligodendrocytes. Okay? So oligodendrocytes electrically insulate neurons. You all can see the models of neurons around here. Okay? So I want you to understand that when signal goes through the neuron, through the cell, the signal is electric. That's it. It's electricity. And you want to insulate the electricity to prevent electricity from leaking out of the neuron, quite literally leaking out. Now, all these cells, so far, that I mentioned, are present only in central nervous system. Satellite cells, which you can see here, okay, satellite cells, present in the peripheral nervous system, and they play the role similar to astrocytes. So they also maintain the ionic concentrations around neurons and, take care of them, but in peripheral nervous system. And finally, there's a Schwann cell. Schwann cell also provides electrical insulation, okay, but in the peripheral nervous system. Now, will I ask you to match the name of the cell and its function? I absolutely will. Will I ask you to identify a cell based on the picture? No. No, stop attributing to Jesus things that I do. It's my work, okay? All right. Does that make sense? So, go ahead.
The Schwann and the satellite are
in peripheral, yes. And if you think, why do we even care? It turns out that Schwann cells and oligodendrocytes, while they perform the same function, then they provide electrical insulation to axons. They are slightly functionally different, and that slight, quote-unquote, slight difference explains why you can reattach a severed arm and it will become functional, but if somebody gets their spine transected, they will be paralyzed. And we'll talk about it. So this is a neuron. So neuron is the transmitting cell in the CNS. And PNS as well, but we're focusing on central, whatever. So parts that you need to know. These branched parts are dendrites, exactly. These dendrites are receptive regions, okay? This part is soma, okay? Also known as a body of a neuron. It can be receptive, but for the most part, when you think about soma, my statement is not absolutely correct, but it's a good way to think about soma. This is where inputs are integrated. because you can imagine that these dendrites can receive signals from many other neurons, so multiple signals at the same time. When the signal is integrated, it proceeds through the axon, yes. So axons are entirely transmitting parts of a neuron. Okay, good. You may notice that the signal goes in one direction to, show you the importance of that. I'm going to use the different color. What the hell? Okay. This is the direction. Dendrites, soma. Usually we mention that little bottleneck called axonal hillock. Then axon, and at the very end you have axon terminals. Okay? Good? So you can see that we go from unidirectional signal. Remember how we talked about I mean this. Hold on a second. Okay, this. Remember how we were talking about the direction of a signal starting with receptor and only in one direction, always one direction, right? Okay, so now you can see that at the cellular level, it's also true. So what is, neurons can be of a different type, structural, okay? The most common are multipolar neurons. You will have a question to match the description of a neuron with its kind. So multipolar neurons have multiple dendrites, one axon, many axon terminals. Neurons do not have more than one axon. There's only one axon. There can be many terminals. This is bipolar neuron. So you have a dendrite, single dendrite that leads to the body, single axon, and multiple terminals as well. And finally, this is a unipolar. So you can see that the cell body is on the side, and dendrites and the axon are basically fused together. So the body is kind of not in the way. Make sense? You do not need to know which neurons are more abundant where. I'm telling you this, but I'm not going to ask that. So like multipolar, a lot of thinking stuff and motor stuff. Bipolar, special senses for the main part. Special senses include sight, hearing, taste, and smell. Unipolar are general senses, such as touch, temperature, and pain. Okay? And yes, you absolutely must know the order of in which order the signal goes through a neuron. Okay? That make sense? I totally will ask this. So now we're going to introduce the concept of a synapse and say a few words about synapses. Okay? So what is a synapse? Synapse is the place where two cells interact, generally speaking. Okay? Period. Two cells interact, not necessarily physically touching each other. So here you can see the synapses between the axon of one neuron and the dendrites of another, so axodendritic. Here you see synapses between the axon of one neuron and the body, or soma, of another, so axosomatic. And here, even synapses can be formed at the very beginning of axon, on axonal helix. Okay? All good so far? So in the nervous system in general, you will see two types of synapses. So one, synapses in general can convey signals in different directions, but the only type of a synapse that can transmit signals in both directions is the electrical synapse. So electrical synapse can convey the signal one way and then another. Why? Because electrical synapses are essentially ion channels that connect two cells. And as you can expect, I have a great analogy for you. So imagine that this door right there, the entry door into the class, is propped open. Okay? Good? And this is one cell, our room, and the hallway is another. And somebody releases a monstrous fart in the hallway, right next to the door. Will we smell it? Yes. So if everybody is in the hallway and somebody releases a monstrous fart in that room, people in the hallway will sense it too. This door, an example of bidirectional signal transmission, basically electrical synapse. Does that make sense? Ions flow one way, ions flow another way. Okay? Now, this is a chemical synapse, and from now on we're only going to talk about chemical synapses. At a chemical synapse, one cell communicates with another by, well, releasing chemicals called neurotransmitters. Chemical synapse is exclusively unidirectional. Is that clear? Unidirectional. So look at this. The signal will go from that cell, from the top cell, to the bottom cell. So while in general synapses can have a signal in both directions, but that's true only for electrical. Chemical synapses will go only one direction. And while we are at it, I'm going to make a very quick remark. I'm not going to be asking that question. I'm going to tell. We're going to talk a lot about electricity. Nothing complicated, just electrical impulse that goes through a neuron, right? I want you to understand a simple thing. Electrical flow, electricity, is a movement of charged particles. It can be charged negatively, it can be charged positively, but if they move, we can say that there is an electrical current. And this is the only time pretty much when we talk about an electrical synapse. We're not even going to mention it anymore. Every time we talk about communication between neurons, you need to think, okay, we have electrical signal in the neuron, and then chemical signal between the neurons. Sounds good? So let's figure out what happens during the synaptic transmission through the chemical synapse. So, these red lines represent an electrical signal. Okay? It's called action potential. We're going to figure out what it is right now. Just trust me, it is a blast of electricity. When this blast of electricity reaches this axon terminal, right? It opens, that blast of electricity literally opens this sodium channels. And you can see that sodium ions start to diffuse from outside of the cell into the inside. Right? A question for you. Why? Why do they diffuse in specifically that direction? Diffuse. Go on. Not the charge. What is diffusion? Movement of chemicals from say again? Nope. From high to low. High concentration to low concentration. Think of a diffusion as a ball going down the slope. You don't need to apply any energy. And the ball will go in the direction of the slope. Clear? Now, if you need to put something uphill, do you need to apply some energy? Yes. So that's going to be your active transport. Now, this is not an example. This is really important. This is the cell. A cell, whatever. Inside of the cell, cytoplasm, a lot of water, a lot of ions. Outside of the cell is extracellular fluid, a lot of water, a lot of ions. You must know the relative concentrations of 4 ions. By relative concentrations, I don't mean numbers. I mean the levels, like which ion has greater concentration on what side of the cell membrane. On the outside, you need to know that sodium, chloride, and calcium have concentrations that are greater outside of the cell. Potassium has a concentration that is greater on the inside. All 4 ions, I will ask about that. You must know this information. I don't care about any other ions. Seriously. You will have a table with all those other ions. Anything but these 4, I don't really care. So again, I actually put it here. So you see they are outside of the cell. Sodium, chloride, and calcium, they are outside, so the concentrations are greater there. So we figured it's because of the concentrations that calcium diffuses in, right? Yes, excellent. Calcium is a great cation, and it signals to these vesicles that I circle around to approach the end of the terminal and to release neurotransmitter, this individual molecules. They are released between the cells into what's called a synaptic cleft. So they diffuse, okay? They diffuse, and eventually they bind to the receptors. It should be your, I call it a 2 a.m. knowledge. When somebody wakes you up at 2 a.m., you barely know who you are, but you're able to tell me like those 4 ions. Or the fact that if we have a chemical, it's going to bind to a receptor, period. Neurotransmitters always bind to some kind of a receptor. Sounds good so far? Awesome. And that's, in a nutshell, that's it. That's it of a transmission. Because we don't think so far, like we're going to say a few words about that. But we don't discuss really what happens in the postsynaptic neuron, okay? So this neuron, this one is presynaptic, before synapse. And this neuron is postsynaptic, after synapse. That's not the end of the story. Think about a synaptic transmission as someone rings your doorbell. Normal people, how do they ring the doorbell? They push it and they release it, because if they hold their finger on, they're psychopaths, right? So you want to stop the transmission. You don't need the transmission to happen all the time. So think about this. If neurotransmitter this neurotransmitter, stays in the cleft, it's going to keep ringing that bell, it's going to keep binding to the receptors, and it's going to keep perpetuating the signal, which is not great. Not at all, because it's going to exhaust postsynaptic neurons. Postsynaptic neuron is going to run out of steam. Okay? So you need to remove neurotransmitter. You have three options. One option is reuptake. You see what happens? The neurotransmitter is taken back into the presynaptic neuron. Okay? Another option is degraded. Okay, not this, sorry. This. Break it. Reasonable, right? Diffusion is also an option, but it sucks because it's slow. Okay? Now, here's what I'm going to do. I'm not going to talk right now about the importance of degradation. We will have a chance to talk about it next week. This is the last thing that I want to highlight, is that there are two types of receptors. That too, but we're not there yet. We're not there yet. We're kind of there. Yeah, actually, you're right. So two types of receptors. Ionotropic, type 1. So see that red hexagon, which is the neurotransmitter. Okay, this hexagon binds to the receptor, and this opens the ion channel. And ions can diffuse. I'm going to check if you're following what I'm saying. What will determine the direction in which a particular ion diffuses? Concentration gradient. If you open a receptor that lets sodium in, right? Well, that lets sodium somewhere. If it's open for sodium. You see what I'm saying? Then sodium will always diffuse into the cell because its concentration outside is greater. If you open a receptor for chloride, it will also diffuse into the cell because chloride concentration outside is also greater. If you open, like if it's a receptor that opens the channel for potassium, potassium will diffuse out because its concentration is greater inside, so it will go out towards its lower concentration. Does that make sense to you? Now, when sodium diffuses into the cell, what type of charge, what vibe does it bring? What type of charge? Positive, because it's positively charged. This is the positive charge. When you receive positive news, are you excited? Yeah. The influx of positive charges means that the cell is being excited. Yes, excitation. Okay? Good? If you bring in negative charges, when you receive negative news, are you like bummed out? Yes. So, negative means that the cell is inhibited. Yeah. Now, when you let potassium leave, does cell become more positive or more negative? But that positive charges are leaving the cell, so it becomes more negative. So, both chloride and potassium, if you bring chloride in or take potassium out, that will inhibit, that will make cell more negative. Am I making sense with this? Now, another type of transmitters is metabotropic. So it means that there have to be some reactions. So a neurotransmitter, when it binds to the receptor, it activates so-called G protein, and this G protein activates some other protein, and that some other protein makes what's called a second messenger. Okay? Good? So, folks, if you don't murder me by the end of the semester for all my analogies, I will be very grateful, but I do have an analogy for that. So, imagine that I need to get into the lab space. I have my card, I go to that lock, swipe the card, doors open. That's ionotropic receptor. It's very quick. Okay, make sense? Imagine you forget something in this room and you need to get back and you need to grab it. You don't have access. So you email me, I email Michaela, Michaela comes over, opens the door for you. It's going to take a while. Does that make sense? In this example, Michaela will be a second messenger. The door will be opened, but it's going to take a while. Generally speaking, ionotropic receptors produce fast responses that do not last long. Metabotropic receptors usually produce slow responses that can last pretty long. Okay? Does that make sense? And questions? No? All good? Okay. So we're going to talk about neurotransmitters, different types of neurotransmitters on Monday. Thank you for being patient with me.